as microbiologists, because not only are you learning, but I'm also learning with you. So I'm excited to, you know, get this quarter going. So now let's get to lab notebook. So I think you've heard this a couple of times already, but I just want to like re-emphasize it that let's see, let me just get out of this mode. All right, so the lab notebook that we want that you're going to be using in our lab is a three ring binder. So that's all we want for you. And don't worry about getting the carbon copy notebooks. We really don't want that. Really, all we want is a three ring binder, hole punch some white paper, put it in there. And I do one for my lab section. I want you to get 6 dividers, whether they're actual dividers or you can get color paper, hole punch them, and use them as dividers. That's totally fine with me. But I want 6 separate sections, and the titles of each section I put them here right here for you. So the first section is going to be called strategies review. So that's going to be a lot of review from your previous courses. Another one is going to be titled benzoate degraders, metallotropes. That's the third one. Winogradsky column sampling, viable cell count, or Winogradsky 8, whichever name you want to use is fine with me, or DYOE, which stands for Do Your Own Enrichment, or Winogradsky B. So label have 6 sections, 6 dividers, and use these labels as labels, and we're going to be adding as we do this the corresponding experiments. But that's all I want. I will send you guys a Word doc describing all my notebook expectations as well. And I just want to do it kind of looks like this. And in there, in that document that I'm going to send you, I put the experiments along with the page numbers where you can find them in the lab notebook, so that you know specifically what goes into each of these sections. That way, it's not a mystery. I'm not trying to trick you. I'm trying to provide you with all the what I expect to find in each section. Now, um, there's one section, section 6, which is going to be something specific for each person. It's going to be different, where you get to choose one of these 5 organisms or group of organisms. So for this experiment, you don't have to worry about all 5. You only worry about the one that you're actually doing. And I put the page numbers corresponding to each group of organisms. So if you happen to get spirochetes, you don't even have to worry about including in your lab notebook any of the other 4. But for the quiz purposes, you do have to know information about all of them. But for the purpose of lab notebook, you only need to include what you choose to do. And we're gonna choose later in the coder, so you don't have to worry about it. Now. I just wanted to make sure that you know about that. So for each of the 6 sections, I you're going to include the following. You're going to want to have an introduction, and in the introduction, in simple terms, I could just tell you that to include relevant background information. But I want to give you more detail as to what that means. And what I mean is that I want you to tell me in the introduction where are the organisms in question or of a specific experiment typically found. Uh, do you typically find them associated to plants? Do you find them in soil? Do you find them in marine environments? For example, if one of the sections was caffeine degradation, which is not, but just as an example, caffeine degraders, you typically find them in soil or associated to plants. So I would say that in the introduction. Another thing that you could include, or that I want you to include, is how is a particular carbon or nitrogen source typically introduced into the environment? Again, is it some of the carbon sources are um, released by as exudates by from plant roots to attract certain bacteria, so they're introduced by plants. Others are contaminants from consumers. Others are metabolites from another bacteria that broke down another metabolite. So tell me how they are introduced. For example, if I was doing caffeine degradation. Caffeine is typically, you know, introduced as a waste product from tea production. They sometimes people use it as fertilizer, and that's how it gets introduced. A lot of people dump their energy drinks or caffeine coffee down the drain. That's another way that it gets introduced into the environment. So tell me, for each of your sections, for each introduction, uh, how is the particular carbon and nitrogen source introduced into the environment? Um, I also want general information of the on the pathway that we're studying. So if we're studying benzoate degradation, like tell me about the pathway that bacteria or the different microorganisms use to break down caffeine or benzoate, whatever that section is. Tell me about that pathway. Is there only one pathway? Are there multiple pathways? So that's the type of information that I'm expecting from that. What genes are involved in that pathway? Do we have one, two, three, four, five genes? What are the you don't have to name every gene if the pathway is long, but give me an example of a gene. For example, if caffeine degraders, if you're looking at the endimethylase pathway, the genes are all called endimethylases. The only difference is that endimethylase A, B, C, D, or F. So tell me a little bit about at least one gene that's part of that pathway. Uh, in some instances, there's not much known about the pathway, so there might not be any known genes or characterized genes. But tell me that there haven't been any characterized yet. Uh, else you can also include in the introduction phenotypic or genotypic characteristics of the organism being studied. So uh, are is the organism, do they form colonies that are pink pigmented? Uh, uh, like what kind of carbons do they grow on? Things like that. That's another aspect that I'm looking for in an introduction. And lastly, I also want you to include strategy. What strategy are we using in the experiment to isolate the target organism? Are we using, direct isolation? Are we using an enrichment strategy? Are we using a selection? So what are we doing in the lab to isolate the target organism? So I'm providing you an example of what is not a good introduction. For example, in the wet mount exercise, the students wants just give me a sentence. So it said wet mounts are used to observe cells. Cell motility and size can be determined. So that really doesn't tell me anything, and like that's pretty much the title of in the lab manual. So that really doesn't show that that person put any effort or didn't tell me any of the points that I just outlined. And now I want to give you an example of a better introduction. Um, the nitrifying bacteria. So you're actually nobody's doing this quarter, so that's why I'm giving you this example. So this person said the nitrifying bacteria are found in diverse lineages of bacteria and archaea. They are commonly found in soil, so that's telling me where the organisms are found in marine environments or sediments, and they are used in wastewater treatment systems for bioremediation. They can make up anywhere from 10 to 15% of the population in these communities. Since they are found in so many lineages, they can be utilized a broad range of inorganic and organic compounds as carbon and energy sources. So tell me about carbon and energy sources. I think that what I would add there examples of what carbon and energy sources. So that would be maybe you know thing that I would criticize about this introduction. And then this person said the main enzymes that they rely on is nitric reductase. So it's telling me about a gene or a gene product, which is only present in anoxic conditions to begin denitrifying from nitrate to nitrogen gas. And then many are part of the gamma, beta, delta, and epsilon protobacteria and firmicutes. So that's it kind of touches a lot of the points that I outlined. And then it says the purpose of the is to isolate anaerobic denitrifiers from Winnigrassy column using different combinations of media and inoculations. So I guess that the only thing that this person didn't put is what's the strategy? Is this person using an enrichment, selective enrichment, direct isolation? So it's a very strong introduction. Some missing pieces, but I think that you get the idea. Now, methods. Some people call it materials and procedures. I like to call it methods section. Um, but here, what I want you to do is to form a sketch or diagram outlining the main steps of the project or the experiment. Don't just say refer to manual page whatever. I don't want that. I want you to do more than that. Include media that is used in each main step. I want you to in some experiments you have to derive or come up with your own media recipe. So I want you to include that. Include incubation times and temperatures. That's very important. And also, what's the source of inoculum? What's the source of bacteria? Are you using soil? Are you using leaves? Are you using the green grassy column? Where is your bacteria source coming from? And if you did something different than what the manual told you to do, I want you to also list those as deviations or things that you did differently in the experiment. So let me give you a quick example. So this is kind of like a sketch or a quick diagram of if you were to do a capillary or a chemotaxis assay. So I provided the media that I was using in each main step, the incubation times, the optical densities that I grew the cells on, um, kind of if I was spinning the cells, what RPMs and for how long, different washing steps, and all the way to the results. So this is kind of like a quick a sketch, so that you have a feeling as to what this looks like. Because I want all of this not to be typed. I want you to be handwritten. This is an example of one of my previous students did. So here, this person says experiments were performed as described in the lab manual, provides page numbers, with the exception that microcentrifuge tube was used instead of the glass tube. So this told me like, hey, the lab manual told me to use glass tube, but I didn't use that. I used microcentrifuge tubes. So you need to list things that you did differently. So most people just say that this. They tell me the page numbers, the lab manual, but I just if you just provide me with that, you're not gonna get a good grade in this portion of the lab notebook. You have to do like a quick sketch of what the experiment or the workflow of the experiment. So for this experiment, this person shows like a Ziploc bag with soil in it and the serial dilution that this person did and which media they plated, the incubation time. But not just that, but any figure or sketch needs to come with a description of what, not a detailed description, a brief description of what this figure represents or what's going on in this figure. So again, get in the habit of making a little sketch or a workflow of the experimental steps. And the reason why I do this is to train you or to help you get in the mindset of planning ahead. Because in some labs, you're going to have to make your own media. And if you don't plan ahead, if you come and you need a certain media and you haven't made it, then you're going to get behind. So I want to encourage you to constantly be working on this. So then the next section is the results section or daily logs. So a lot of the labs that we're going to be doing in this course are go throughout the whole quarter. So it's not just on Tuesday and that's it. You never do anything with it again. So there's experiments that we're going to start on Thursday and they're going to go on for like 3 or 4 or 5 lab periods. There's some that are going to go throughout the whole quarter. So you need to make sure that you make logs of everything that you do and the dates. So the first thing I want you to include is obviously dates. You need to date every entry, whether that's a drawing, an observation, any notes, and everything needs to be dated. Write the name of the organisms in correct scientific format. What that means is the um, genus needs to be capitalized, species lowercase, and it needs to be either italicized or underlined. So make sure that you do everything in the appropriate scientific format. All drawings, picture, tables, graphs, they need to have a legend or a description, right? If you just provide me a table and I don't know what it is or what it represents, you need to tell me what I'm looking at. Otherwise, it's meaning. It has no meaning actually. So make sure you do that. I want you to provide the total magnification of anything that you view under the microscope, but not just total magnification, but what type of microscopy you're using. Did you use bright field, or did you use phase contrast? Did you use dark field? Tell me what microscopy you used to view your specimens. Now, everything needs to be written in pen. There's no the only exceptions is if you make a drawing, you can use color pencil or pencil. That's totally okay. But everything else, the introduction, uh, it needs to be not typewritten, written in hand, handwritten. Also, the conclusion, the results, everything needs to be handwritten, with the exception with drawings or sketches. You can hand draw that, as well, or you can use pencils. But everything else needs to be in pen. Um, Here's an example from one of my previous students. You can see that this experiment went on for at least 14 days, and this person wrote the day number of that experiment and the date. That's very key, to the date. Uh, I tend to color code everything that I do. So I usually use a red pen for dates. I use maybe a black pen for notes, a blue pen for something else. So you can color code things too. I'm not requiring you to color code things, but it will be not only help me, but it will also help you find information as well. So you can see that this person used color pencils. It's totally fine. It's a drawing, but it told me that this was a wet mount, came from enrichment, was a chitin degrader. It told me that it was a 100x objective lens magnification under phase contrast 3. Told me a little bit of what that person observed. That person observed flaming motility, gave me dimensions of the cells that this person observed, and it came with a little note or description of what was going on in here. So that's the kind of level of detail that I'm looking for. And not only that, this person told me also incubation temperature. The only thing that this person didn't provide was probably the incubation, how long what they did incubate for, or stuff like that. So, but you get the idea. Um, Now, conclusions. In the conclusion, what I want you to include is what did you learn about from performing that specific experiment? And also, like, I also want you to go back and interpret the data and the results that you gather, not just list the data. I want you to interpret it. And also, what experiments can you follow up your findings? Like, what would you do next if you had time? Based on your observations and your data, what will be the future directions of this experiment? So here you had to use, maybe do some reading, or, use your imagination, as to what will be an experiment to follow up. There are times in which, an target organism is difficult to isolate in the time frame that we have, and some of you might not get anything, but but just because you don't get anything doesn't mean that you're going to leave this section blank. What I want you to talk about in that case is reasons why you were not able to isolate anything. Were the cells growing more slowly than predicted? You know, what was the temperature too high, too low? Give me ideas as to, or predict reasons why something fell, why the experiment fell, and also provide possible ways to troubleshoot. How will you overcome the speed bumps that you encounter in the experiments? Now, for the last section that I want you to include for each lab that you're going to be putting in your lab notebook is, uh, questions. They are often found at the end of the lab, but they are sometimes appear in the middle of the lab or within the procedure. So make sure that you, as you read, that you note questions. What I do, I have a highlighter, and as I'm reading, every time I encounter a question, I highlight it in green or blue, whichever color you want. That way, once you're done with the lab, you can always go back and just visually spot check which questions you need to go back and answer. So make sure that you do that. And now here's my email. Feel free to email me if you have any questions or if something was not clear. I am happy to, be in contact with you with email, or if you prefer to do a small, quick Zoom meeting, I'm also open for that. I just want to also tell you that while you can contact me in Canvas, I'm quicker to respond if you email me to my UC Davis email. There's a lag of when you send me a message through Canvas. It takes, I don't know why, but it takes sometimes a couple of hours for them to notify me that I have a message. So, but my email notifies me immediately. So if you want a quicker response, just use my email. I check Canvas once or twice a day. So like if you message me through Canvas, I might take longer to it may take me longer to give you a response. So just be aware of that. All right, so that's the end of Lab Notebook